Hi everyone, please can you hear me? Assalamu alaikum. I can't share my screen. Hello, can you hear me? Please, you can type in the chat box if you can hear me.
Yes, doctor, we can hear you.
Yes, I can't share my screen. It's been disabled.
Is there
So
any
that,
PA present in the...
yeah, I'm looking at the name of the participant. So far, it's only the students. I've sent him an email. Okay. Okay, I understand Dr. Oban is still giving you the lectures.
Probably,
Okay.
yes, he did not finish it.
Really?
Yeah.
I thought it was 5 minutes to the time. That's why. Okay. So we hold on. Hi everyone. Do you think I should start since the lecture slides are on Moodle? Or wait approval for screen sharing? What do you think? Hello? Hello? Guys, can you? Yeah. So do you think we can start? You just go on to Moodle and you follow the slide presentation as I move on. Is that fine with everyone? Okay. So we wait 5 minutes. Thank you.
Hello, doctor. We just finished
Yes.
the other lecture now. I made you a co-host now, so you can share your screen.
Thank you so much.
Thank you, doctor.
So, assalamu alaikum, everyone. I'm going to be discussing infectious diarrheal agents, starting with bacterial agents. By the end of today's discussion, these are what I expected of you. To have an idea of the epidemiology, in which area, in what part of the world do you have infectious diarrheal agents as a predominant or prevalent cause of diarrhea, the mode of acquisition of the agent, and the mechanism of disease, because the mechanism affects the presentation of the patient. So it's very important that for each agent that we are going to discuss in this lecture and subsequent lectures, that you know these mechanisms. When do you send a stool sample for laboratory evaluation? Because it's not in every case of diarrhea that the stool must get to the lab. So when do you make up your mind as a doctor that I'm going to evaluate this patient further? Then what samples are you going to send and how are you going to collect the samples? What happens to the sample in the lab? And then when can the lab tell you, no, this is not a good sample, you have to send another one. So the World Health Organization defines diarrhea as the passing of liquid or watery stools at least 3 times in a 24-hour period. The key word is the fact that the stools are watery or they are in a liquid form. So the consistency is more important than the frequency. When you have blood or very mucoid stool, we refer to that as dysentery as opposed to just ordinary diarrhea. Now, I want you to know that diarrhea is a clinical presentation. You can have both infective and non-infective causes. For example, a patient with thyrotoxicosis may have diarrhea as part of the presentation, and it's not an infective process. So, like I said, I'm going to focus on bacterial agents that are agents of diarrhea. But be that as it may, you know that when we talk of microbes, it includes bacteria, viral, and parasitic agents. And for each group of microbe, there are very common agents that can result in diarrhea. For example, with bacterial agents, common organisms include Escherichia coli, the ones that are referred to as diarrheagenic, as opposed to other E. coli that can cause UTI or that can cause sepsis. We have Campylobacter jejuni, and the other organism on the list, and viruses. For example, rotavirus, which is a very common viral agent of infection in young children, like kids under 2 years. Rotavirus is the leading agent of diarrhea in these agents. We have norovirus, which is associated with cruise ships. So when the patient comes to you and gives you the history that I just returned from a cruise tour and the patient presents with diarrhea, norovirus should come to mind. We also have protozoan agents, parasitic agents, protozoan and helminths that are associated with diarrhea. So what is the epidemiology like? Diarrheal diseases are a big problem in developing countries. Death in children below the age of 5 years. And it also contributes to malnutrition in this group of kids. And this is because a lot of nutrients are lost in the diarrhea stool. There is ineffective and poor healthcare facility support for these patients. And a study has shown that in developing countries, diarrhea is responsible for about 1 death every 10 seconds in children under 5 years old. So here you have a map that was adapted from data from WHO. And like we said, just a minute. Okay. Diarrhea diseases are a big problem in developing countries, particularly countries in Southeast Asia, African countries, where you have poor water supply, poor personal hygiene, poor management of waste disposal, because most of these agents that cause the diarrhea are passed out in the feces of an index infected patient. So where you have this poor infrastructure, these organisms are prevalent in these regions. How is the infection acquired? It's mainly fecal-oral. This could be through contaminated food or water. However, with some agents, you can have person-to-person transfer, as with Shigella, with Entamoeba E. coli, where you have people who have sex, men who have sex with men. You can have a person-to-person transfer of these agents. You can also have fomites playing a role with agents such as rotavirus and Giardia lamblia. So let's look at each agent. Oh, okay, before that, clinical presentation. Now, the mechanism of disease usually affects clinical presentation. So patients may present with acute watery diarrhea, in which the stools are essentially liquid. There is no blood, and you have just a few mucus secretion in the stool sample. And this is because here, the basic mechanism of disease is that of lack of electrolyte reabsorption. So you have a sort of osmotic diarrhea, or at times you can have a secretory diarrhea. In some patients, they present with dysentery. So you have the clinical impression that you are dealing with an invasive diarrhea. It means the agent can compromise the integrity of these enterocytes. As a result, you may have bloody stools or dysentery. An example of organisms that can cause this is listed there. At times, you may have patient presenting with persistent diarrhea. The diarrhea has been on for more than 14 days. It's just, nothing can be done in terms of first aid at home. to resolve the diarrhea. So the patient eventually presents to the clinic and they give you a very long history. Parasitic agents such as Giardia can cause this. So let's discuss some specific agents with emphasis on the mechanism of diarrhea and how you can make a diagnosis in the lab. We start with Vibrio cholerae because this is an agent that can cause pandemic as well as endemic disease. In fact, there have been 7 recorded pandemic episodes of cholera outbreak, and we'll discuss the particular biotype and serotypes that are responsible for infection. So Vibrio cholerae is a gram-negative rod, but it has a curved appearance, so it looks like a comma when we gram stain. It has a single polar flagellum and it is highly motile. The organism is fermentative, it is a facultative anaerobe, and it is halophilic, that is, it requires high sodium chloride content for subdivided into over 200 serogroups based on the somatic wall antigen, that is the O antigen. If you still remember from your POD, with gram negatives, we have the O antigen, right? If the organism is motile, we have the H antigen for flagellum. And if the organism has a capsule, we also have the capsular antigen. So with Vibrio cholerae, the O antigen is more useful in terms of serotyping. It's much more useful than the H antigen because the H antigen seems to be common to all serogroups, so it becomes difficult to differentiate. Vibrio cholerae serogroup 01 is further subdivided into serotypes Inaba, Ogawa, and Hikojima. Now, I'm not going to go too much into the details. What I just want you to know is that these serotypes are based on laboratory characteristics, okay? And it's possible for these strains to interchange. So you can have Inaba transiting through Hikojima to Ogawa, and vice versa, that is Ogawa strains transiting through Hikojima back to Inaba. But like I said, I'm not going to bother you too much. You don't need the details. 0 is further biotyped into classical and now the 7 pandemics that have been recorded up to date, 6 out of it was associated with the classical 01 Vibrio cholerae. The current pandemic is associated with L-TOR. Now, L-TOR appears to be less virulent than the classical in terms of the disease presentation. Now, cholera, that is the disease caused by this organism, is mediated by an enterotoxin that is produced by this organism when it colonizes the enterocyte. So the cholera toxin and the pilus that is required to colonize the intestinal epithelial cells are regarded as virulence factors. Non-toxin producing strains of Vibrio cholerae are not pathogenic. So like I said earlier, Vibrio cholerae 01 is responsible for 6 out of the 7 pandemics that have been recorded so far. There's another serotype that seems to cause cholera, but it is not as virulent as serotype 01, and that's 0139. Other serotypes of Vibrio cholerae are associated with disease conditions such as wound infection, sepsis, and what have you. Most of them don't seem to cause cholera. So cholera is associated with serotype 01 and 0139. This organism is associated with fresh bodies of water where you have high salt content, and it's associated with chitinous shellfish. The organism can live a non-parasitic life by living and multiplying freely in the environment. It's when they are ingested that then they can cause problem in the patient. During the warm weather, the rate of replication appears to increase. Most infection is acquired through the consumption of freshly contaminated water. And that's why we said these agents are prevalent in areas where you have poor sanitation, environmental sanitation. So when you have an infected patient defecating in the river where everyone comes to fetch water for use, then this encourages the spread of the organism in a community. Like I said, direct person-to-person spread is rare. And because the organism is highly sensitive to pH, at very low pH, at acidic pH, the organism is destroyed. So you need a high infectious dose before the infection can be established. So it's only the organisms that survive the stomach acid that will establish infection in the GI, because the pH in the GI is not acidic. So what is the pathogenesis like? The hallmark is the effect of the toxin on the enterocyte. Cholera toxin is an enterotoxin. It is different from a cytotoxin. An enterotoxin will affect the function of the enterocyte. So instead of absorption, you're going to start having secretion. That's an enterotoxin. A cytotoxin will destroy the entire cell. It can either lead to apoptosis or it can lead to membrane rupture of the enterocyte. So that's the basic difference between an enterocyte and enterotoxin rather, and a cytotoxin. So let's look at what happens with cholera. When the organism colonizes the epithelial cells, okay, it starts to produce the toxin. Now, the toxin produced by cholera toxin is described as an AB toxin. An AB toxin means you have two units, the B and the A subunit. The B subunit is for binding to the receptors on the enterocytes. In this case, the receptors are the ganglioside GM1. Now, when this happens, endocytosis is triggered and the toxin moves in. The toxin is eventually presented to the endoplasmic reticulum, where you will have the A subunit, which is made up of A1 and A2 units that are held together by a sulfide bond. When this unit gets to the endoplasmic reticulum, the A1 subunit is eventually cleaved off. This A1 unit is an enzyme that catalyzes the transfer of ADP ribose from NAD onto the G protein. The effect of this is to activate the production of cyclic adenosine monophosphate, that is cyclic AMP. When the concentration of cyclic AMP increases, the resultant effect on the enterocyte is to activate channels that will allow chloride ion to escape from the enterocyte. Other electrolytes will follow to balance out, as well as water, because these are osmotically active. Now, apart from allowing the chloride channels to be activated, the effect of the enterocyte, of the enterotoxin on the enterocyte is also to prevent the absorption of sodium chloride from the lumen into the enterocyte. So the net result is massive production of watery stool. So that's why we say that the stools are profuse and watery. In terms of diagnosis, cholera is usually diagnosed clinically by the time you see a patient presenting and the stool being passed looks like this, you know that you are dealing with a case of cholera. More so if you have an outbreak taking place in the community at that time. So this is referred to as the rice water stool. Now, there is another rare presentation of cholera in which you don't have massive production of stool. This is referred to as cholera Zika. And in this case, you have paralysis of the ileum, so you have massive production of watery secretions into the lumen of the small intestine, but it is not being passed out. So you're going to have abdominal distension from massive outpouring of this fluid and electrolyte. So when the patient presents, because the patient is not passing stools, you may misdiagnose the patient. And that's why this particular condition is associated with high mortality. Because before you realize what's going on, the patient has practically lost all the body fluid volume into the intestines. Now, let me say that you do not need to send the stool sample to the lab to make a diagnosis for cholera. It is mainly clinical. However, from the public health standpoint, you need to get the sample to the lab for epidemiological reasons. So what do we look out for in the lab? Like I said, these are gram-negative, curved shaped, comma-shaped bacilli, as you can see in the micrograph, and they are motile with one flagellum at one end. It is oxidase positive. This is very important because it helps to distinguish it from members of Enterobacteriaceae. And you can actually isolate the organism over a wide temperature range between 10 degrees to 37 degrees centigrade. Another laboratory feature is the appearance of Vibrio cholerae on the selective medium thiosulfate citrate bile salt sucrose agar, TCBS for short. This medium will help differentiate Vibrio cholerae from other Vibrio species. And this is because Vibrio cholerae can ferment sucrose. That's why you have the sucrose in the name. The fermentation of sucrose results in acidic end products that will change the color of the indicator in the medium to yellow. So when you see yellow colonies, you know that you're dealing with Vibrio cholerae. Other Vibrio species will appear greenish because they don't ferment sucrose, and you need to do further laboratory tests to identify them. You can also culture this organism on McConkey, but it's going to appear as a non-lactose fermenter. So you need to go a step further and perform other biochemical tests. Now, let me say that in a non-endemic area, we don't usually use the TCBS to isolate cholera because it's not cost effective. It is an expensive medium. So where you don't have an outbreak, where it's not an endemic disease, you still go for McConkey and then you perform other laboratory tests. Remember, I told you that the virulence factors associated with this organism is the toxin, the enterotoxin it produces, and the pili that is required for attachment to the epithelium. So these are all encoded for by genes, which we can actually detect in an isolated isolated organism. So we can actually perform molecular assays to detect the presence of the CTX gene or the TCPA gene So if they are present, we know we are dealing with a pathogenic strain of Vibrio cholerae. We now move on to Escherichia coli. Escherichia coli is now an organism you know very well. We discussed E. coli when we were discussing UTI. So this is an organism that is a little bit complex. The strains that cause UTI are different from the strains that cause diarrhea. The strains that cause diarrhea are classified based on the syndrome associated with each strain or based on the serotype. That is, we are using the somatic and the flagellar antigens to characterize them. Recognized strains involved in diarrhea diseases include the enterohemorrhagic E. coli, which is also referred to, it's a subset actually, of Shiga toxin producing Escherichia coli. These are also referred to as verotoxin producing Escherichia coli. We have the enteroinvasive E. coli, the enteropathogenic E. coli, the enterotoxigenic E. coli, enteroaggregative, and then we have the adherent invasive Escherichia coli. And we are going to discuss them so that you can understand why they are distinct entities. So diarrhea-causing strains of E. coli have specific fimbrial antigen that enhance colonization. Remember that in the disease process, colonization is the first step. If the organism cannot attach to the site, it cannot reproduce, it cannot cause infection. So these antigens, these febrile antigens enhance colonization and adherence to the mucosa of the small intestine. Like I said earlier, we have different strains that exhibit different pathogenic mechanisms in the disease process. So let's discuss them into details. ETEC stands for enterotoxigenic E. coli. And like the name suggests, the main mechanism is enterotoxin production. Remember, I told you an enterotoxin is different from a cytotoxin. ETEC is the leading cause of traveler's diarrhea and diarrhea in children in developing countries. The toxins are plasmid encoded. You know what plasmids are. They are extra chromosomal genetic materials. They tend to confer on microbes a survival advantage. And unfortunately, in some cases, they may also bear antimicrobial resistant genes and pathogenicity genes. In this case, that's what's happening here. The toxin is the virulence factor. The two types of toxins that have been associated with this particular strain of E. coli are the heat labile, the LT, and the heat stable, ST genes. Now, things are so complex that you can have some strains producing just one of these genes, of these toxins, or you can have some strains producing both. One thing you should note is that the heat labile toxin is similar in structure and action to cholera toxin. So that means you have an AB toxin, which when it is activated results in the production of cyclic AMP with the net result of chloride secretion and inhibition of sodium chloride absorption as well. So I won't go over the LT because I've explained that with the cholera toxin, but the ST, the mechanism of pathogenesis is a little bit different. This tends to bind to a membrane spanning enzyme. That is, this enzyme has a membrane component and an intracellular component, but it's still the same one molecule. And that membrane spanning enzyme is called the guanylate cyclase, or C, or GCC. GCC can be found at the apical membrane of intestinal epithelial cells, and binding of the toxin to the extracellular domain stimulates intracellular enzymatic activity. The resultant effect here is the production of cyclic GMP. Now, with cyclic GMP, the net result is also the secretion of chloride ions, and other ions follow passively as well as water, and it also prevents the reabsorption of sodium chloride. So the net result is production of watery diarrhea. Now, in this situation with cholera, with enterotoxigenic E. coli, you notice that the epithelial cells are not destroyed in any way. It's only the function that is observed that is affected. So you don't have bloody diarrhea. The patient may present with abdominal cramps. The patient may have fever. At times, there may be or may not be vomiting. But the point is, there is no blood in the stool, okay? And you don't have an overt inflammatory response, so you won't see white blood cells in the stool. With enteropathogenic E. coli, the organism is associated with infant diarrhea. It's prevalent in children below the age of 1 year, okay? Mostly in developing countries. When you have E. coli, enteropathogenic E. coli causing outbreaks in developed countries, it's usually associated with infant nurseries, okay? It's not a prevalent environmental condition. Now, the hallmark of infection due to enteropathogenic E. coli is what is called the attaching and effacing signs or histopathological features. And this can be seen on biopsy specimen. So what happens is when the organism attaches to the the villi, it introduces various toxins that results in the loss of microvilli. Remember that the microvilli observe absorptive function. So instead of absorption taking place, because these microvilli have been destroyed, that process no longer occurs, and then you have diarrhea. Here you have a micrograph of a patient who had infection due to EPEC, and as you can see the pedestal, you can see that the microvilli have been completely effaced where you have attachment of the organism. So that's why we say it's an attachment effacement pathological sign. We move on to the enterohemorrhagic, which is a subset of Shiga toxin producing E. coli. Now from the name, you have an idea that this is something similar to the Shigella toxin. Actually, E. coli species that produce Shiga toxin are somehow related to Shigella, but they are not Shigella. They just have some close similarities. This organism can cause diarrhea in infants and children. The impact and importance of this particular strain of infection, particularly in public health domain, is immense because it is the main cause of acute renal failure in children in many countries. The prototype organism is E. coli 0157H7. The O, it's not zero. The O stands for somatic antigen. The H stands for flagella antigen. So it was the first to be linked to hemorrhagic colitis and hemolytic uremic syndrome in the early 1980s. And the common feature among this particular subset of E. coli is the ability to produce Shiga toxin. So there are different types of Shiga toxin based on the molecular structure. You have Shiga toxin 1 and Shiga toxin 2. Even at that, there are subsets of each of these sugar toxins. So you have sugar toxin 1A, 1B, sugar toxin 2A, 2B, and things like that. Now, the effect of the sugar toxin is to inhibit protein synthesis. So this is not affecting the electrolyte function, absorption, or secretion, but actually it is affecting protein synthesis at the level of the ribosomal RNA. Okay? So the sugar toxin can also affect cell signal transduction and immune modulation, causing pro-inflammatory and pro-apoptotic responses. So it's possible when stool samples of patients with infection due to this particular strain, when they get to the lab, that we can see some white blood cells because you have some degree of pro-inflammatory activities going on. The classic intestinal pathology characteristic of E. coli 015787 infection includes hemorrhage and edema in the lamina propria. So here you have the diagram explaining the effect of the sugar toxin. It binds to GB3. GB3 is a glycolipid that is associated with the enterocyte. And with that, the toxin is internalized, okay? And eventually it gets to the endoplasmic reticulum through the Golgi complex and acts at the level of the ribosomal RNA, eventually leading to inhibition of protein synthesis and eventually cell death. So with enterohemorrhagic, because you can have cell death, you can have blood in the stool. So apart from having inflammatory cells, we can see inflammatory cells, the stools may also be hemorrhagic. Now, the receptor GB3 that is present on the enterocyte is also present in high concentrations in human renal tissue. And this partly explains why you have some renal damage as well. So although the problem is in the GI, the kidneys are affected because the receptors are also found in the kidneys and also in the brain. So at times you may also have some CNS component to the clinical presentation. Unlike the effect on in the CNS, when this toxin, enterotoxin, binds to human renal endothelial cells, it has a cytotoxic effect. It eventually destroys the renal cells. And sugar toxin type 2 is more important in the development of hemolytic uremic syndrome. We move on to enteroaggregative E. coli. Here, the infection tends to be self-limiting. However, some patients may develop long-lasting diarrhea lasting more than 14 days. The pathogenesis is not well understood. The diarrhea associated is often watery with the presence of mucus with or without blood, with abdominal cramps, vomiting, and low-grade fever. So what happens here? The organism aggregates, okay? Planktonic means that they are free living, they are not attached to anything yet. Then they attach to the intestinal epithelial cells, resulting in colonization. When they start to divide, what they do is they form a biofilm. The biofilm eventually disrupts the function of the epithelial cells and their integrity. That's why at times you may have blood in the stool, because by the time the epithelial cells dies off, there is sloughing and then there is bleeding. So release of Bacterial toxin induces damage and you have increased secretion because the integrity of the enterocytes have been compromised. Enteroinvasive Escherichia coli is the causative agent of dysentery. This is invasive. The name has already given out the pathogenic mechanism. So it is the causative agent of dysentery in humans, especially in developing countries. Diarrhea due to Escherichia E. coli and Shigella is caused by the invasion and penetration of bacteria into the enterocytes, leading to their destruction. Now, despite the similarities in invasion mechanisms and symptoms of the disease, the dose of EC is much higher than that of Shigella. Shigella is about the most infectious of all these infectious agents we are going to discuss. You need just a little number of organisms to establish infection. Pathogenesis of E. coli suggests that its features are virtually identical to that of Shigella. The current model of Shigella and enteroinvasive E. coli pathogenesis comprises some steps. First, you have colonization, then epithelial cell penetration, then there is lysis of the endocytic vacuole, the escape, and then there's intracellular multiplication. Then there is cell-to-cell spread through directional movement from one cytoplasm of an infected cell to the non-adjacent non-infected cell. So what do I mean? This is a model for Shigella enteroinvasive infections, epithelial cells, colon. When they invade the epithelium, it's usually through the M cell, from what studies have shown. Now, after reaching the epithelium and they invade the epithelial cells, they are phagocytosed as usual by resident macrophages. But what the organism does is to resist destruction by the macrophage. Instead, they activate apoptotic mechanisms in the macrophage. So the macrophage will lyse and release the organism on the intracellular side. What happens then is that the organism is picked up, and within the cell, a lot of biochemical reactions that result in cytoskeletal rearrangement, giving the organism, because the organism is non-motile, it gives it a tail to propel it to the adjacent non-infected cell. So you can see it's no longer coming in from the lumen. It's now coming from the subluminal surface, and you have infection of the adjacent cell. Now, at times, after the infection has been established, you can have invasion from the lumen because of the monocytes that are attracted to the site of infection. So you can actually have some extracellular transfer of the organism to the subluminal surface. Then we have adherent invasive Escherichia coli. Now, this particular strain of E. coli is associated with irritable bowel syndrome, for example, Crohn's disease. The pathogenesis is not well understood, but it is known that resected segments of intestine from patients with Crohn can lead to the culture of this particular subset of E. coli. So they are particularly prevalent in patients with irritable bowel disease. Now, the AIEC pathotype is defined by the ability to adhere to and invade epithelial cells. They replicate within epithelial cells and macrophages, so they don't destroy the macrophages. Studies on adherent invasive E. coli show that it is able to adhere to intestinal mucosa by binding to the carcinoembryonic antigen-related cell adhesion molecule 6. invade intestinal epithelial cells by using the host cell active microfilaments and microtubules. It replicates intracellularly, translocates across human intestinal barrier and move into deeper tissues. So here you have a diagram explaining the process. Again, it appears that the M cells are important in the access of this organism to the subluminal surfaces. And then you have different pro-inflammatory cytokines involved in the production of the disease. And just like we explained with entero-invasive, you can have adjacent cell-to-cell transfer of organisms. Okay, so you can see that we've gone through the different prototypes of organisms, E. coli organisms, and they are not the same in terms of the way they cause infection. So what is the approach to diagnosis? Definitely, one size cannot fit all. So you have to look at various approaches like phenotypic and molecular typing, serotypings, and other nucleic acid amplification techniques, and other gel assays that we can perform. Then you can also use the newer technologies like the MALDI-TOF, which stands for Matrix Assisted Laser Desorption Ionization Time of Flight. Now, this is like a mass spectrophotometry, okay? Then you can also perform sequencing, whole genome sequencing, or high throughput. These are newer techniques that are being used in the approach to diagnosis. Now, one thing I want you to note is that unlike the other E. coli we have discussed, particularly the one causing UTI, the diarrhea causing E. coli may not be lactose fermenting. Remember, we've always told you that E. coli is lactose fermenting, but with strains that cause diarrhea, about 90% of them are lactose fermenting. The remaining 10% are actually non-lactose fermenting. So in this case, the indole reaction is very important. Over 99% of diarrhea-causing E. coli strains are indole positive, okay? Now, with E. coli 0157H7, it is important because this is associated with hemolytic uremic syndrome. It is important to detect it early. So a highly selective medium that can help you to quickly identify this in a stool sample is the sorbitol McConkey agar. With the other strains that are not 0157H7, sorbitol is fermented, so you have pinkish colonies. However, with the particular strain, 0157H7, it is sorbitol negative, that is, it doesn't ferment sorbitol. So it shows like colorless colonies when you culture the stool sample. So this is very important for you to note. So let's discuss quickly why we need to perform a laboratory analysis. Before you send a stool sample to the lab, you have to ask yourself these questions. Will the identification of the pathogen influence the way I'm going to manage this patient? If your answer is no, then you don't need to send it to the lab. But if your answer is yes, it's very important it gets to the lab. Is the identification of the causative agent important from a public health perspective? For example, the serotype, say for example, you have an outbreak of cholera, you know your diagnosis is clinical, but you need to send the sample to the lab. Yeah, you have to send it to the lab. Are we sure we are dealing with 01 or 0139? So things like that. So these are the questions you should ask before sending the sample to the lab. Now, What are the indications? Testing should be considered when your patient presents with diarrhea of more than a day's duration that is associated with bloody stools, because this means you are dealing with an invasive organism. So you want to be sure what is going on. The patient presents with fever, or the patient has symptoms of sepsis. The patient is highly dehydrated. These are all reasons to further strengthen your decision to send the sample to the lab. If the patient has had a recent antibiotic use, because you can have antibiotic-associated diarrhea. If the patient is immunocompromised, for example, a renal transplant patient, a patient with HIV progressing to AIDS, a preterm baby, so you have to do something. The lab needs to help you out. You also send the sample to the lab when identification is important from a public health perspective. Now, in the laboratory, which test can we perform? Now, it depends on your patient population. If you want to employ somebody in a fast food restaurant, culture is important. Screening for cysts of Giardia and crypto is important. Looking out for ova of parasites is also important. In children less than 5 years, culture is very important. So I don't have to go through everything. The plus sign indicates that you must carry out that test based on the patient population. Now, when do we perform stool culture? We perform it when we want to detect bacterial pathogenic organisms, because you can't culture viruses readily. That is going to take a long time. So the aim is you are thinking of a bacterial pathogen. So you want us to identify which agent are we dealing with. The types of specimens that can be sent for culture include stool. In patients who are very, very compromised, that is, maybe they are unconscious, or maybe they have some sort of neurological problems that makes them fecally incontinent, you may want to send in a rectal swab or a biopsy. The important thing, if you are going to do this, is to make sure that you place the swab or the biopsy in a transport medium. If you don't do that, it's going to dry up, and then we won't isolate anything in the lab. So you'll be taught the acceptable transport medium when you have your lab. You can also send in duodenal colostomy, ileostomy, or sigmoid aspirate. These are in patients who have had some form of surgery to divert intestinal contents, okay? Maybe there is a tumor somewhere that is obstructing the GI, so they have to do some form of diversion. So depending on the surgery that has been performed, you may have to actually aspirate from the duodenum or the colostomy. Colostomy means that you've made an opening connecting to the large intestine. Now, who will collect the specimen? If the patient is okay, the patient is more mentally competent, can understand instructions, give the sterile container to the patient, instruct the patient. Now, if stool is not obtainable from the patient by the patient, the nursing staff or the physician will have to intervene. Now, the quantity of specimen, well, I find this quite how should I say, funny, because we don't expect you to go weighing. So this is what the book says, but I guess in practice, what you just do is that you must get a sizable amount of stool sample to the lab. But the book says at least 5G of feces. So we don't expect the patient to go weighing the samples. When does the lab reject a sample? When there is inappropriate or poor quality specimen sent to the lab. For example, we just said that consistency is important in the definition of diarrhea. So we are saying that the stool is watery or in the liquid form. So don't send a well-formed stool to the lab and you are saying that you are investigating diarrhea. It doesn't make sense, okay? So the lab will not process that sample. The lab can process formed stool under certain conditions, but definitely not when you say you are investigating diarrhea. Now, when you send a sample to the lab and you are not specific, as you can see, I've already told you that the approach to diagnosis is varied. So you cannot expect the laboratory personnel to start making up the diagnosis for you. So there's nothing like please culture for everything. As you find out, different organisms, different media, different incubation periods. So what exactly are you screening for? If you're thinking of E. coli, then we won't be using TCBS because we know that's for Vibrio cholerae. If you're thinking of cholera, we won't be thinking of Sorbitol McConkey. Okay? So if you don't give us an idea, then we can't process the sample. If the sample lands in the lab and there is no label and there is no requisition form, it's going to be rejected. If the sample lands to the lab and the collection time indicated on the sample bottle shows that the sample has lasted for like 4 hours before getting to the lab, then it's not going to be processed. You'll get to know why this is important when you have your lab. If the specimen container is contaminated or leaking, then the lab is going to reject that sample because if it can get out, something can get in. Now you should know that stool samples should be examined, as soon as possible after collection, okay? And stool specimen cools, the drop in pH will inhibit some organisms. So in conclusion, different disease mechanisms are deployed by different bacterial agents, and it's important you know this. Only adequately collected and transported specimens are acceptable for laboratory analysis when needed. That's the important thing, when needed. So we've gone through the reasons why at times you have to send the stool sample to the lab. Now, good personal, food and environmental hygiene will prevent and control infectious diarrhea in a community. So thank you. Any questions? Please do not send this type of stool sample to the lab. Any questions? Check the chat box. If you have any question, I guess you can unmute yourself or you can type it in the chat box. Are we good?
Thank you, doctor.
Yes. No questions?
There is no question in the chat.
Okay. Thank you so much. Enjoy the rest of the day.
Thank you,
Bye.
doctor. Bye.
Okay.